You can't just stand there and stare at me. Go sit down. Do you want another bone? I'll give you another bone. You have to be a good girl. There you go. Calm down. That's not going to work, but it makes me feel better. Okay. Welcome to the genetics review. Okay. It's not alive. Stop yelling at it. Just eat it. She's probably going to interrupt me every time I start talking until she settles down. Okay. I don't know. We'll see. I'll try. I might have to put her downstairs, but then she'll scream at me downstairs. So we'll try to get through this. Anyway. Okay. Our final exam is in Magic Card 3006. This is our regular room. So if I messed that up, I don't think I did, but is our regular room where we always meet on Wednesday, December 14th at 10:15 in the morning. It's a 2-hour cumulative exam. All 9 modules are represented equally. So 9 because we did not get through Mod 10, so that is not on the exam. As I mentioned in class on Wednesday of last week, I gave you some practice problem, or I gave you some topics to practice for the 4 open response questions. So you are getting 4 open response, but the same amount of multiple choice, true, false, and fill in that I gave you before. So you're being given an extra 45 minutes to complete 1 additional question, which is the open response question. In class, we discussed what are those questions going to be. So one of them, so we certainly did the chi-square quite a few times. We did it 3 different ways that chi-square was represented. So I bolded and put that in purple because you should expect to see some form of a chi-square on the exam, definitely. I also bolded this, the lac operon, because you have not been tested on it yet. So you will definitely get a lac operon question as an open response question on the exam. So you can just definitely practice those because you know you're going to need it. It's definitely going to be on the test. And then you have some options for the remaining 2 questions. So one is a chi-square in some form, one is the lac operon. So you'll have to know how to draw it. You'll have to know how it works. So how is it kept off and how do you turn it on? And then you'll have to be able to do one of, and then the remaining 2 open response questions will be one of these types of questions. It could be interpret a pedigree, which means you need to know how to understand the symbols so that you can draw or interpret the pedigree, excuse me. You need to know how to do a mutational analysis. So that means you have to either start with DNA, go to RNA, so that you can determine what your amino acid is. That's gene expression. So that means you need to know how to transcribe and translate a DNA molecule. You should also know how to go from amino acid back to DNA to some extent. So there's too much redundancy in the genetic code for you to perfectly predict what a genetic sequence is given an amino acid sequence, but you should be able to predict what types of changes likely happened at the genetic level to produce an amino acid mutation that you're looking at. So you need to know how to, so those I think correlate to questions 2 and 3 on the problem set 7, module 7. For the population genetics calculations, we did a migration question on the last exam. We also did a testing for Hardy-Weinberg equilibrium. But what if I decide to do one of these other 2 options for the chi-square and not that? That means you need some other, you could potentially have some other type of pop-gen question on there. And so it's either not going to have a pop-gen question in open response, or it will be an inbreeding question, which you'll find examples in the study guide for Mod 8, which I'm going to review today. Or also one of the problem set questions for Mod 8 was an inbreeding question as well. Oh crap, am I recording this? Yes, okay. Okay, and then it could also be some kind of chromosome counting during the different cell cycle stages. This is like open response question number 3 in the first exam, where you're given a particular haploid number and you have to know how many chromosomes are there in G1, S, G2, or M, different stages of M, and also how many DNA molecules there are. And so that is essentially, if you have 8 chromosomes, where's my pen? If you have 8 chromosomes and you are in G1, you still have 8 chromosomes when you exit S phase into G2, but you have double the genetic content because during S phase DNA duplicates. So if you have 8 chromosomes in G1, you still have 8 chromosomes in G2, but 16 DNA molecules in G2 because each single chromatid is now represented as a pair of sister chromatids. All right, so so for today's review, I did come up with questions to address each of those potential open response. So we'll demo, practice all of those things in today's review. But first, let's talk a little bit about how you study for the rest of the exam, besides the open response. So I'll help you with that, but let's talk about what you can focus on for making sure you understand the multiple choice, true, false, and fill-in expectations. There are 20 total, so 2 for most modules, 2 questions total across all of these. And then 3 for two of the modules will have 3 questions represented. Anything from this list that did not make it into the open response will be converted into a type of multiple choice question. So you still need to know how to do these things because you need to be able to recognize them in the form of a multiple choice question. So for example, if I don't do a mutational analysis question, I can very easily make one into a multiple choice by saying, given the codon AUU, it changed from AUU to AUG. What type of mutation is that at the DNA level? Is that a transition, transversion, an indel? Or what type of mutation is that at the protein level? Is it a missense, a nonsense, a silent, or a neutral? So that's one example of how to turn a mutational analysis into a multiple choice. So you can practice that too. How would she ask this as a multiple choice question? Try to come up with some on your own or work in group study to each of you come up with multiple choice questions. Another one could be given that the 2N chromosome count in G1 equals 8. What is the 2N chromosome count in G2? And you would say 8, it's also 8. Or how many DNA molecules are there in a cell in G2, given that the diploid number is 8? It would be 16 DNA molecules, right? Or given this picture, would you predict that this is a cell in mitosis, meiosis 1, or meiosis 2? I'd have to pick a very specific phase so that you could actually distinguish those 3 different M phases. Okay, so try to turn anything from the above, try to turn those things into some kind of multiple choice or true false or something like that to help you study that, or just make sure you study those topics. And then, so for the terminology, focus on your study guides for all kinds of concepts. If you've been doing your study guides as you go, you've got a nice stack of paper that you can sort of start to run through. And any place within those study guides that you either don't remember or you had to like, oh yeah, that's right, I totally forgot about that thing. You might want to go a little deeper and practice it as a problem set. Go to the exam where I asked you about it and practice those questions. Go a little bit deeper into that module to clarify any of those things that you didn't get the first time around or you didn't get on your review. Module 2, 3, 7, and 9 are pretty heavy on terms. And so if you need to make sure you master the terminology because you need to be able to speak the language in order to apply the concept, make sure you focus on mods 2, 3, 7, and 9 for terminology, because that's where most of the terminology hits you is in those 4 modules. Review your past exams. I have now made all of the exam answer keys in blank exams available on Blackboard. I believe it's under, I don't remember. Let me go look. Oh, it's easy. It's exam blanks and answer keys. You're going to find them all there. All three exams are represented. This does mean you will probably not, actually, in fact, you definitely will not see the same question twice, because you're getting all of them here. But it is a good idea to review them because you will get similar questions as you have seen before. You can use those blank exams to test yourself. So before you review the content for, say, Unit 1, take the Unit 1 exam again and see how you do. Keep note of how long it takes you also, so you can try to practice your time management also. And then if you want to practice the format of multiple choice and true false, go back and review the unit quizzes on Blackboard. I did make them all available, so all of these unit quizzes are currently available for you to review through them. I am not accepting any other additional assignments at this point, which is why I felt comfortable enough to open those up so you can review them as much as you want. And then use the adaptive quizzes on Achieve. So I also did that. Well, I don't know if I can log into Achieve from here. This is my old computer. So what I did here is I went to our genetics class. I went through module by module, and I made it so that your adaptive quizzes were available again all the way through December 18th. So you can go in here and you can, oh, I don't think it shows me in instructor view. Yeah, it doesn't show me an instructor view what the actual quiz looks like. But you can go through and retake those adaptive quizzes. It'll keep throwing questions at you. So I would go through each of the modules and spend like 10 minutes or so flipping through and using the adaptive quizzes to help you practice multiple choice formats. I do use the book pretty heavily in this class, so the adaptive quizzes should be accurate to what you need to know for the exam. Okay, any questions before I start talking about chi-squares? Drinking water and coffee at the same time. I'm hoping that balances out. Is that how it works? Let's say that's how it works. Okay. All right. So let's move into the review part. Okay, so I'm going to make this easier to see. I'm going to activate my pen. So I recognize that we have used the chi-square in 3 different ways. And so let's talk a little bit about how those 3 ways are similar and how very specifically they are different as well. So in general, these different points define a chi-square. So a chi-square test in some form is what you need to know. So this is what I told you last week. This is what you need to know. You need to know how to do a goodness of fit test. And in order to run a goodness of fit test, in order to determine what your expected values are, you're going to need to know how to do a monohybrid cross to do that. So that would be something like this crossed with this. That's a monohybrid cross. That's 1 gene, 2 alleles. You also need to know how to apply the chi-square to do a gene linkage test, a test for gene linkage, which means you are required to know how to do a dihybrid cross in order to generate your expected results. And your dihybrid cross is similar to this, only you're looking at 2 genes. So it's something like this. That's a dihybrid cross. We also use the chi-square to measure observed and expected allele and genotype frequencies when we were testing for Hardy-Weinberg equilibrium. So you need to know how to apply the p squared plus 2pq plus q squared equals 1 Hardy-Weinberg equilibrium equation to determine what your expected frequencies are. So let's walk through, I'll walk you through each of those processes and some demonstrations. But first, let's focus on what is similar across all of these things. What makes all these chi-squares the same? What are some of the same principles we apply across all of them? And so this might help clarify why we use chi-squares to answer these questions. So the question we are testing in every single case is, are observed values significantly different from expected values? That is why the chi-square equation is observed minus expected. So that's the difference between your observed and expected values, normalized to your expected values. This is one chi-square test, or one chi-square application, but typically for every diploid population, we're looking at multiple observations. So we're looking at brown eyes versus blue eyes, or white fur versus orange fur. So we usually have multiple observations, and therefore we would have multiple expected values. And so normally what we have to do is we have to apply this observed versus expected comparison for one observation, but we also need to do it for any of your other observations that you're looking at also. So you would have to do this for observation 1, which is going to have its own expected value, but you're going to have to add that to the difference between observed and expected values for your other observation, so observation 2. And so that's why when we normally write this, we write it as the sum of those values. Will we get all the questions on the finals we've been given? All the equations? Oh, certainly, yeah. Anything that I gave you previously as an appendix, so your genetic code table, your equation bank, the chi-square table, and your amino acid table, so that you can determine if something is a neutral mutation, you'll get all of that stuff. Yep, no problem. I'm gonna, I've actually already made that, so I'll post that to Blackboard so you have it in advance. Somebody did ask, do we get a cheat sheet in this class like we do in biochem? No, you don't. Okay. The general, okay, so now let's see. Oh, okay, fine. So the general null hypothesis, anytime you ask this question, are observed values significantly different from expected values? Your null hypothesis in general is, stop. In general is, any difference you see between observed and expected values, any difference you see is due to chance and no other reason. That's it. That's your null hypothesis for all of these. Differences between observed and expected values is due to chance. We just tend to make your null hypothesis a little bit more specific for the very specific question you are asking. And we use the chi-square test to ask 3 different types of questions, the 3 bullets that you see above. So I'll go into more detail in a moment. This means you can interpret your general null hypothesis by looking at your p-value. You're going to get a chi-square, you have to calculate a degree of freedom, and using those 2 values, you get a p-value. And if your p-value is less than 0.05, your values are different for some kind of reason, and you would reject your null hypothesis. If your p-value is greater than 0.05, your values are not different, and variation is due to chance, and you would accept your null hypothesis that any differences are due to chance. It's just that these hypotheses can be made specific depending on what type of values you're looking at and one type of concept you're exploring. So let's start. Oh, and so then you would, again, you get your chi-square value. Your chi-square values are all of these black values in between, but you also have a degree of freedom associated with each of your chi-square tests, which can be calculated in different ways, but almost always comes out to 1 for a diploid organisms, which that's all we've been really studying, except for bacteria, which is why we don't apply the chi-square in that one. It's a little bit more complicated, and I don't want to make things even more complicated than they already are. So if you got a chi-square value of 3, a chi-square value of 3 falls between these 2 chi-square values, and so your p-value would be bigger than 0.05, so p is greater than 0.05, but p is less than 0.1. And so for that case, for this very specific case, you are above the 0.05 threshold, which means they are not different, which means you accept your null hypothesis. If your chi-square value is 7, that is going to fall down here between these 2 chi-square values, if your chi-square value is 7. And so that means your p-value is greater than 0.005, but less than 0.01. And so because we are below the 0.05 threshold, they are different. They are definitely different. And you would reject that null hypothesis. Okay, so let's break it down by chi-square test. The first time you apply this, it was the test of simple dominance. And so the chi-square test of simple dominance, which is the goodness of fit test. So this is your goodness of fit test. And so your specific null hypothesis, we can adjust the general null hypothesis, which is any difference between observed and expected values is due to chance and no other reason. But we can adjust that to be a little bit more specific for the type of question we're asking, which is this gene inherited by simple dominance? So your null hypothesis for this specific question of simple dominance is any difference between your observed and expected phenotypes controlled by a single gene is due to chance and no other reason. So if your P is less than 0.05 and we reject our null hypothesis, that means your values are different because the genes are not inherited by simple dominance. If your P value is greater than 0.05 and we accept the null hypothesis, this means that the values are not different. The variation is due to chance because the genes are inherited by simple dominance. Okay. So how do you get your expected values? You would have to do a monohybrid cross, just like Mendel would, a Punnett square, some type of monohybrid cross where you're looking at one gene, two alleles, and a diploid species that is one gene, two alleles, and three possible genotypes. This will help you determine your expected phenotype ratios because Mendel will tell you what you expect to see percentage-wise, and then you apply that to the number of individuals you've got working with, your n value. Your degree of freedom here is calculated as the number of observable phenotypes minus 1. So let's apply. Fruit flies can either have black eyes or red eyes, and that is controlled by the eye color gene. My face is going to get real close and very awkward on the screen because I need to bring the computer closer to me to write, so no judgments, please. Okay. So fruit flies can either have black eyes or red eyes, which is controlled by the eye color gene. Two fruit flies mate, and one is heterozygous for the eye color gene, and the other is homozygous for the B allele. And we produce 46 progeny. 31 have black eyes, and the rest have red eyes. Oh, what I didn't tell you is black eyes are dominant over red eyes. Sorry about that. Okay. In order to interpret the cross that you have to do, you have to know the language. You need to know what heterozygous for the eye color gene means and what homozygous for the B allele, the little b allele means. And so heterozygous for the eye color gene means one of our flies is heterozygous. It has one copy of each allele. The other fly is homozygous for little b. So that's what our other fly looks like. Our N is 46. We have 46 total flies, and 31 of them have black eyes, and the rest have red eyes. So that would be 46 minus 31. I'm going to need my calculator for this. Not for this, but for the whole. Hold on. Okay. So with 31 have black eyes and the rest have red eyes, that means 15 have red eyes. Okay. Is this eye color trait inherited by simple dominance? So we have to apply our goodness of fit test here. So we have our observed data. These are observations, the observed number of black flies, the observed number of red flies. So this is O1, our first observation. This is O2, our other observation. But in order to apply the chi-square equation, we need our expected values, right? So how do we determine our expected values? To do that, you would act like Mendel and try to predict what's going to happen given this cross. given what you know about simple dominance. And so they are expected values because we are assuming our null hypothesis is correct, that these are inherited by simple dominance. So given that our null hypothesis is correct, we expect to see %50 heterozygous and 50% homozygous for the little b allele. So that means 2 out of 4 are going to have red eyes, and 2 out of 4 are going to have, oh, whoops. We're going to have black eyes because that's what this genotype will give, and 2 out of 4 are going to have red eyes because that's what this genotype will give. So given that our N is 46, we expect half of that 46, or 23, to be black-eyed individual flies, and 23 to be red-eyed flies. So those are your those are your expected values for each observation. This being your first expected value and this being your other expected value. So observed minus expected over expected, observed minus expected over expected. And let's not forget to square the numerator here. Okay, so let me give myself a bit more space. Okay. So we've got our 2 observations here. Bet you my pen is not active. Okay, and this color's rough. I love red. It's just a little tough to read. Okay. So for our first observation, which would be the black-eyed flies, our chi-square equals, we observe 31 of them, as was given to us in the problem. We expect 23 of them based on Mendel's laws. We square that and we divide this by 23. Giving us 2.78. Our other observation is red eyes. So we need to do this for red eyes too. We are observing 15 red eye individuals. We expect 23 of them, given Mendel's laws of simple dominance and assuming our null hypothesis is correct. Oh, interesting. Okay. Also 2.78. Let me just do that again, just to make sure. Yep, okay. And so to get the total chi-square, that means we have to add these 2 values together. 2.78 plus 2.78 gives us 5.56. Our degree of freedom, remember from the last slide, is df equals the number of observable phenotypes. That's 2 minus 1. We have 2 phenotypes, black and red. So 2 minus 1, or 1. So we have a chi-square value of 5.56, a df of 1. Let's head to our chi-square table. Let's actually all right. Get my pen back. Our df is 1. So we are restricted only to the chi-square values in this top row, all of these black values here in the top row. And what did I say? It was 5.56. So 5.56 is between here. So this means our p-value is greater than 0.01, but less than 0.005. We are below the 0.05 mark. So we would reject our null hypothesis that they are inherited by simple dominance. Clearly, there's something else influencing their pattern of inheritance here, because otherwise expected and observed values would be so similar that would be way up here, and the chi-square value would be very small. Because the chi-square value is large, that pushes us down below the 0.05 threshold. We reject that it's being inherited by simple dominance. It's got to be inherited by some other method. One of the others that we talked about, like codominance or incomplete dominance or lethal alleles or X linkage or something like that. One of the exceptions that we talked about in Mod 5. Okay, any questions so far about that? All right. Now let's apply the test for independent assortment. So another way that we applied Mendel's laws, or we tried to, or we looked at, use the chi-square to verify if Mendel was correct, was this test for gene linkage. Mendel says everything independently assorts during meiosis. So this is Mendel's null hypothesis, that any difference between an observed and expected phenotypes controlled by two genes is due to chance and for no other reason. So if p is less than 0.05, you reject. Your values are different because the genes are linked and they don't independently assort during meiosis. So we reject the null hypothesis. However, if the p-value is greater than 0.05, the values are not significantly different. Variation is just due to chance because the genes are not linked and they do assort independently during meiosis. That's your null hypothesis, right? So you would accept the null hypothesis if your p-value is greater than 0.05. This means that if your genes are linked and don't assort independently, the only way to separate them is to recombine them. Because the only way that you would get gene B separated from gene C is if there was a recombination event on the sister chromosome between those two regions. And the only way that you're going to get a recombination event between those two regions is if they're fairly, at least a little bit far apart. So that a recombination event is likely to occur between them. The only way to separate two linked alleles is to do recombination, which is going to happen less than 50% of the time. So under 50% of your progeny will be recombinant progeny. If they are not linked, that means they're super far apart on the same chromosome, like this. So a recombination event is virtually guaranteed because there's so much space, or they are simply on different chromosomes. So instead of being very far apart, one's on chromosome 1 and the other is on chromosome 2. And so they assort independently during meiosis. So they're either on different chromosomes so far apart that recombination is guaranteed, or they're on totally different chromosomes. In order to get your expected values here, you need to do a dihybrid cross, just like Mendel would do, to predict what your expected ratios would be. And then you can apply those ratios to a number of individuals that you're dealing with in your population to determine what your expected numbers should be, given that everything is independently assorting. Our degree of freedom is a little bit different. It's still the number of observable phenotypes minus 1, but because we have two genes, and they are not linked to each other, we have to look at the two genes separately. So the number of observable phenotypes for gene A minus 1 and the number of observable phenotypes for gene B minus 1. So your DF is a little bit different. Again, it's probably going to equal 1 because for any particular diploid gene locus, we usually have two observable phenotypes. Let's apply. Okay. Fruit flies can either have black or red eyes. So same example, it's controlled by the eye color gene B, black is dominant over red. Fruit flies also have either smooth or wrinkled wings, which is controlled by the wing texture gene T. Smooth is dominant over wrinkles. Now we have two genes, a B gene and a T gene. Two fruit flies mate. One is heterozygous for the both genes, for both genes, sorry, for both genes. And the other is homozygous for little b and heterozygous for the T gene. Again, you need to know what the language is. You need to be able to interpret this reading comprehension in order to start with the correct parents, the correct cross. So one that is heterozygous for both genes, that means it's going to be heterozygous for B and also heterozygous for T. And one that is homozygous for B and heterozygous for T looks like this. So here's our two parents. They produce 46 progeny, so I kept N the same. Okay, and now we have 4 possible genotypes because we're looking at 2 genes. So instead of having 3 possible genotypes, heterozygous, homozygous, black, homozygous, red, we can combine each of those 3 possibilities in different ways to give us 4 possible genotypic combinations: black eyes with smooth wings, black eyes with wrinkled wings, or red eyes with smooth wings and red eyes with wrinkled wings. So here's our observed data. So we have black eyes, smooth wings, that's 14. Black eyes and wrinkled wings, 11. We have red eyes and smooth wings, that's 11. So I'm just representing phenotypes here. These could also be gametes because there's only 1 copy of each gene. And then we have red eyes and wrinkled wings, which is 10. So that's our observable data. In order to get our expected data, however, we are going to need to do a dihybrid cross. All right. Oh, I forgot to mention one thing at the top of the review. I will mention it in a moment. Okay. All right. So in order to do this dihybrid cross, I like to do branch diagrams for dihybrid cross because it keeps your ratios straight. You don't have to count up cells in a 16 square Punnett square. If it's something like a heterozygote for everything crossed with heterozygote for everything, you're going to generate 16. Lots of them will be unique. So rather than that, you can consider each gene separately first and what that looks like in a Punnett square and then apply it here. So for this, cross with this, we actually just did this on the previous page. It's 1/2 heterozygote and 1/2 homozygote recessive. For a heterozygote crossed with another heterozygote, that is your 1/4. And I'm memorizing these a little bit. You can do the Punnett square if you want, but we've done these crosses so many times at the beginning of the semester that it's just kind of in there now. But this would be, you know, your Punnett square that looks like this. And that's where these ratios are coming from. And this one would be your Punnett square that looks like this, and that's where those ratios are coming from. Okay, so I'm going to start with 1/2 heterozygote for the black and white for the eye color gene, and then 1/2 homozygous recessive for the eye color gene, and then each of those gets one of the 3 possibilities for wing texture genotype. I have to text my husband. He thinks I am dead because I have not spoken to him since he left this morning. It's funny to think I'm dead. I haven't left the house. I don't know why I'd be dead. Gas leak, I guess. Yeah, I'm just going to tell him I'm good. Okay. All right, so for each of these, we now multiply the ratios with each other to get what we would expect to see if heterozygote B and also homozygous big T were to find each other in the same cell or be joined in the same cell. So 1/2 times 1/4 is 1/8. There's a 1/8 probability that this would happen in the same cell. There's a 1/2 probability that this would happen in the same cell. And then just keep applying that concept as you move down the diagram. Okay, so there's your expected genotype ratios. We can't use those because some of these genotypes are going to have the same phenotype. So let's see, let's apply phenotypes to these. So this would be black eyes and smooth wings. This is also black eyes and smooth wings. So 2 individual flies that have black eyes and smooth wings could be 2 different genotypes. And so what we can do is add the frequency of those 2 genotypes to determine what my overall phenotype ratio would be. And so 1/8 plus 1/4, the common denominator there is 16. 1/8 is 2/16. 1/4 is 4/16. So 2 plus 4 would be 6. Did I do that right? No, wait a second, I messed that up. No, wait. No, I did that right.
Yeah, that's right.
Yep, that's right. Yeah, sorry. Thanks, Tawani. I appreciate the support on that one. She's just like, she's just watching me struggle. Okay. Because I usually do everything out by hand, so it's just tough for me to see it in my head. It's how I learned. So these individuals would have black eyes, but wrinkled wings. And that would be the only one. That's 1/8. Because now we're down here where everybody has red eyes. So red eyes here because they're homozygous recessive for the little b allele, but these 2 individuals have smooth wings. This individual has a wrinkled wing. And so this is also 6/16. Let's make this common denominator across the board, just so it's a little easier. And same here. Okay. So now we have our expected phenotype ratios for each of the possible phenotypic classes. So now I'm gonna make more space for myself, pop a little slide in here. And I'm going to grab those numbers because I'm going to need these numbers here. Because those are my observed values. And now I've got my expected ratios, which are not values yet. They're just ratios right now. Oh, didn't like that. Let's try again. Okay, so we can't put ratios in a chi-square table or a chi-square equation. You have to put numbers in the chi-square equation. And so for both of these, my n equals 46. So these all add up to 46. When we apply our ratios, they should also add to 46. So 6/16 of 46 is 17.25. That's my expected value. I actually have not done this out. I created this problem last night, but I have not actually done it out. So I don't know the answer. 46 times, oh, and this is going to be the same. Okay. So now I have my observed values here for each of my 4 observed phenotype classes. And then I have my expected values here for each of my 4 observed classes. So I now have to do my observed minus expected for each class. So this might be a little bit easier to arrange in a table. So let's try that. So let's see. So we have black, smooth, black wrinkle. I have red. Oh, good. My husband is happy that I am not dead. Always a good thing. I think he always thinks someone's going to come and like break into the house and murder me or something. Like, you'll see another ring. So, you know, it's all right. Okay. Here's our possible classes. And let's do observed minus expected for our first. And then we'll square that for the second one. We'll divide it by expected value here. And then we'll sum it up down here on the bottom. Okay. Oops, I put that in the wrong spot. So for my black smooth individuals, I observe 14. I expect 17.25. For my black wrinkled, I have observed 11. I expect 5.75. For my red smooth, I observe 11, but I expect 17.25. For my red wrinkled, I observe 10. I expect 5.75. So this is negative 3.25. And then when I square that, I get 10.56. Now I'm going to divide it by my expected, which is 17.25, giving me 20. You're in the way. I'm just going to move you over here. 0.61. I think I'm using 2 sig figs this whole time, so I'll just keep doing that. Just be consistent. I don't care how many you use, just be consistent. For the next one, black wrinkled is 11 minus 5.75. That gives me 5.25. I then square that to get 27.56. I then divide that by expected, which is 5.75, to give me 4. Ooh, that's a big one. That already I can tell my chi-square is going to be significantly below 0.05 because that's a pretty big difference right there. But let's keep going. Let's finish the thought. 11 minus 17.25 is negative 6.25. Squaring that gives me 39.06 divided by expected is 2.26. And then for the last phenotypic class, 10 minus 5.75 is 4.25 squared is 18.06 divided by expected is 3.14. And now I got to add all of those up. Giving me a final chi-square of 10.8. my degree of freedom is my number of observable phenotypic classes for gene B, that's black and white. No, black and red, sorry. So that's 2 minus 1. And then I gotta also consider my number of observable phenotype classes for gene T, that is smooth and wrinkled. So that's 2 minus 1. Each of those equals 1, and 1 times 1 is 1. So now let's go to the chi-square table and look for 10.8. Okay. So again, I'm restricted to this top row because my DF is 1. So I can only look at chi-squares in this top row. My chi-square is 8 something, so I'm way down here, which means my p-value is less than 0.005. That's far below the threshold for significance. So that means I reject my null hypothesis that these genes assort independently. They appear to be linked because they differences between expected and observed values are very different, significantly different. And so the genes must be linked and travel together. What that essentially means is we're getting far fewer recombinant genotypes than we expect to see, far fewer recombinant genotypes. So our parents are black eyes and smooth wings, and they're also, so that would be black eyes and smooth wings. And they are also red eyes and smooth wings. That's here. My recombinant would be the other classes, black and wrinkled and red and wrinkled. And so what I just said was we are seeing fewer recombinant. So this one is recombinant and this one is recombinant than we expect to see. Or just they're different. They're different from what we expect to see. And so that makes sense because those are the areas where the chi-square was the biggest, where you see the biggest differences. Oh, break for Christmas. Hold on a second. I got a Christmas clock going every hour. I love it. Pause for Christmas. Okay, enough of the festivities, back to work. All right, so we are now testing for evolution with this other chi-square. Wow, we've been talking about chi-squares for like almost an hour, but let's finish the thought. So the last application of a chi-square test was the test for evolution. So the testing of Hardy-Weinberg equilibrium. Your specific null hypothesis, again, remember your general is any difference between observed and expected phenotypes is due to chance and no other reason. But we can make this a little more specific for testing for Hardy-Weinberg equilibrium by stating any difference between expected and observed alleles, which is essentially phenotypes, right, is due to chance and no other reason. If P is less than 0.05, the values are significantly different because allele frequencies are changing due to one of my 5 assumptions being wrong. Remember, when a population is in Hardy-Weinberg equilibrium, you assume there is no selection, no mutation, no migration, no effective genetic drift, and everybody is randomly mating. But if my p-value here is less than 0.05, this population is engaging in one of these things. So maybe there's selection, or maybe there's mutation, or maybe there's migration. Essentially, one of the assumptions that I make in order for a population to be in equilibrium is wrong if my p is less than 0.05. It is evolving. Allele frequencies are changing. Therefore, allele frequencies cannot be used to predict genotype frequencies, and you cannot use the Hardy-Weinberg equilibrium equation. Can't do it. You have to use observed data. If p is greater than 0.05, that means the values are not significantly different. Any variation we see is due to chance because allele frequencies are in Hardy-Weinberg equilibrium and are not changing. This means my predictions are correct that allele frequencies are not changing. They can be used to predict genotype frequency, and the population is not evolving. I can also assume that all 5 of my assumptions are correct. Observed genotype frequencies can be measured using data, And then expected genotype frequencies can be measured using allele frequencies and the Hardy-Weinberg equilibrium equation. That's how you would get each of those values. So observed genotype frequencies, you can measure from data by taking how many individuals have that genotype divided by the total number of individuals. Expected frequencies can be measured using the Hardy-Weinberg equilibrium equation. Those are different. Our degree of freedom is the number of observable genotypes, which for any diploid organism, 1 gene, 2 alleles is 3 observable genotypes, 2 homozygous classes and 1 heterozygote, and the number of associated alleles, and for any diploid population, that's going to be 2. So the numbers change a little bit in the DF, but 3 minus 2, still 1. So our degree of freedom is still 1 here. Let's test for it. Wow. So we've got a black bear. This is from your study guide. So we've got black bears that are brown or black in color. Occasionally there's a white bear that appear off the coast of British Columbia. We determined that white color coat is a recessive mutation. So these individuals are white. The wild type allele at this locus A encodes black or brown. And so having 2 copies of A gives you black. 1 copy of A gives you brown. Okay. What are the observed frequencies of the A and G alleles in these bears? Okay. So for part A, observed frequencies of alleles. So this is observed allele frequency. Let's first figure out how many genotypes we have. A genotype is an individual. We're diploid populations, unless you're bacteria. So we have 87 bears in this population, 87 genotypes. That means how many alleles do we have? We have 2 times that many because each genotype carries 2 alleles. Okay, so to determine the frequency of the A allele in this population, you have to consider the genotypes that all contain the A allele. Because the homozygous, so because these genotypes contain 2 alleles, there's 42 individuals that carry the AA genotype, and each of those 42 individuals have 2 copies of big A. So we have to count each of those twice. And then we can add the heterozygotes that each carry only 1 copy of A. So you only have to count those once. And then we divide that by the total number of alleles in our population, which is 174. So this means in order to determine and use the chi-square test of Hardy-Weinberg equilibrium, you also need to know how to do genotype and allele frequency calculations. So each of these chi-square tests to determine your expected values requires you understand the concept of genetics. You understand what Mendel's law of simple dominance means and what a monohybrid and how to use a monohybrid cross. You understand what Mendel's law of independent assortment means, and you know how to do a dihybrid cross. You understand what Hardy-Weinberg equilibrium means, and you know how to calculate observed and expected genotype frequencies. So there's multiple skills all embedded within the chi-square. This second part's easy because this A, the amount of A and the amount of G is always going to equal 1 because this is a percentage. So 62% of the population has the big A allele. That means the frequency of the G allele is very easy to calculate as 1 minus 0.62. Because if 62% of the population carries the big A allele, then 38% of the population must carry the G allele. Okay, so now we just calculated allele frequency. And let's label this one P and this one Q. Now part B says, give the expected genotype frequencies. Expected genotype frequencies. So if it is the expected, what we expect to see under Hardy-Weinberg equilibrium, that means we can use the Hardy-Weinberg equilibrium equation and genotype frequencies to predict genotype, allele frequencies to predict genotype frequencies. So let's do that first before I read the rest of B. So that means this is going to be 0.62 squared plus 2 times 0.62 times 0.38 plus 0.38 squared equals 1. So that means the frequency of big A is 0.38. The frequency of the heterozygote is 0.47, and the frequency of homozygous G is 0.14. And make sure you do your check to make sure those are all actually equal 1 so that you can catch any calculation errors. And it's 0.99, close enough. Okay. Now that's my expected genotype frequencies. And if the population is in Hardy-Weinberg equilibrium, I expect to be able to use allele frequencies to predict genotype frequencies. That's one of the predictions of the Hardy-Weinberg law. I have an N of 87, and so I can now figure out the number of big A I expect to see, the number of heterozygotes I expect to see, and the number of big G I expect to see by multiplying each of these percentages by N. So 0.38 times, so 38% of my population is big A, and I have 87 in my population. I expect to see 33.06 bears that are Black. If 47% of my population of 87, I expect to be brown. And 14% of my population of 87 is 12. So I expect 12 of them to be white. Okay, so now I have my observed genotypes. I have my expected genotypes. And so you should sense this coming, right? Because now I have to do my chi-square test. So I'm gonna need a little bit more space for that. Expected, and I'm gonna need my observed. Oh, that's probably not gonna work. 42, 24, 21. Is that right? 42, 24. Oh, 42, 24, 21. And so now I can do my observed minus expected for each of my classes. So what? And we'll use the same observed minus expected squared divided by expected. Okay, so I observed 42. I expected 33.66. For AG, I observed 24. I expect 40.89. For GG, I observed 21. I expect 12. And so let's go through the math. 42 minus 33.66 is 8.34 squared it is 69.56 divided by expected is 2.07. Well, that's a huge number. 285.27 divided by expected. Oh yeah, this population is definitely not in Hardy-Weinberg equilibrium. I can already tell because of how big those individual chi-squares are. So certainly adding them up is going to make it even bigger. It's so much easier to design questions where you're below 0.05. I don't know why. I don't know why. Anyway, so now I'm going to add these up. That's a pretty high chi-square. I have 3 observable genotypes and 2 associated alleles. So my DF is 1. My chi-square is way below this 7.8, so my P is less than 0.005. I reject my null hypothesis. These populations are clearly evolving. Allele frequencies are definitely changing. And so I cannot use them to predict genotype frequencies. The population is not in Hardy-Weinberg equilibrium. One of my 5 assumptions is incorrect. So they're either doing non-random mating, there's significant mutation, there's significant migration, there's significant effects of genetic drift because the population is really small, or there's significant amounts of natural selection. That was all 3. That was like the most intense chi-square breakdown ever. I literally talked about it for a full hour. Questions? Is anybody here? Okay, I still have some people. Like, I'm boring everyone to tears. This is so much. Okay, enough chi-square stuff. Enough. But all these slides should be available to you on Blackboard already. I recommend going through the slides where I break down the questions it's asking, the null hypothesis, what you do with P, what you do with chi-square. Understanding what each of those values means and knowing how to apply the question means, especially the general question, means you can use chi-square to test lots of different differences, right? Observed and expected differences for anything, not just in genetics, but for lots of different types of data. How many times you flip a coin or something. All right, let's talk about the lac operon. For the exam, you need to know how to model the lac operon. What that means is you need to know the basics of the different components of the lac operon and what each of them do. So there are structural genes in the lac operon. These make proteins. And they make proteins that aid in lactose metabolism. And again, the reason why these are organized under a single regulatory element, so all 3 structural proteins are organized under a single promoter and a single operator, is because bacteria have their environments change on them immediately. They go from being out in the environment to being inside of a person when that person consumes a piece of food or whatever. So they need to be able to turn on expression of genes that are important for that change in the environment. Should lactose suddenly become available and there's no glucose around, then they're going to want to turn on all of these proteins right away so that they can access a metabolizable sugar, which is lactose. So these proteins aid in lactose metabolism. There are other proteins that we consider to be regulatory. So they are not binding lactose and breaking it up. They're not allowing lactose into the cell. They're not doing something else important. They are regulatory proteins that either keep the operon off under normal conditions or allow the operon to turn on under abnormal conditions, so the presence of lactose. The only regulatory protein in this system is LAC-I. It binds to the operator and keeps the operon off under normal conditions. There's also an inducer because we need to be able to turn it on somehow. So how do we get these proteins to get it? How do we express these proteins? How do we get gene expression from the LAC operon? In the presence of the inducing molecule, which is allolactose, it destroys the ability of the repressor to do its job. So it deactivates the repressor. And so now the operator is open, the promoter is open, and the operon is induced to turn on. Allolactose is only present when there's lactose in the environment. Then there are regulatory elements, and these are DNA sequences. DNA sequence where something else binds to it to either promote gene expression or inhibit gene expression. That's how these DNA elements regulate something downstream of it that are attached. One is the promoter, and that promoter is where RNA polymerase binds to promote gene expression. So this would be in the on position. The other is the operator, and when something binds to it, which is the LAC-I protein, binds to the operator, gene expression is off. Let's walk it through by drawing it out. So I've got my LAC operon, my DNA, which always draws a box because it's double-stranded. So here's my DNA. I've got a promoter, I've got an operator, and they partially overlap each other so that binding to the operator affects binding at the promoter. So that's why they partially overlap. And then we have our three structural proteins, Y, Z, and A. When lactose is absent, this means it's off. So elsewhere in the genome, LAC-I is under the control of its own promoter. And that's a constitutively expressed gene. So it always makes RNA. It always makes the protein LAC-I under every condition. When there's no lactose, so lactose is absent, that means LAC-I is bound to the operator. And so RNA polymerase cannot bind. There's something in the way, something blocking its ability to bind to the promoter. So we get no gene expression. When lactose is present, a tiny little bit, so let's redraw the operon. And then let's draw our regulatory protein. And it's always making RNA, it's always making protein. But there's allolactose present because that's the inducing molecule. Binds to LAC-I right in the same spot, right in the DNA binding domain, so that it's not able to bind the operator. So now that cannot bind to the operator anymore. This means RNA polymerase is able to bind to the promoter, which promotes gene expression. That means we're going to get RNA made, a single RNA transcript with 3 open reading frames. That's 3 start and 3 stop codons for each of these open reading frames. So we're going to make beta-galactosidase, which will bind to lactose and break it into metabolizable sugars. We're going to make LAC-Y, which allows more lactose to enter the cell, which will keep this on. And then we're going to make LAC-A, which does something important. All right, with me so far, questions? Any questions? Okay. All right, so now let's mess it up. promoter, that guy, promoter, operator, Z, Y, A. You tell me, what should I'm going to mutate one gene. What should it be? P. Brooke says P. Okay, Brooke, you win. So now we're going to mutate P. So now this regulatory element, this DNA sequence where RNA polymerase binds, is mutated. So when that is mutated, that means RNA polymerase is unable to bind in this case. So let's say there's no lactose in the cell. When there's no lactose in the cell, allolactose is not produced. That means the inhibiting repressor molecule binds to the operator. We block RNA polymerase from binding. And so we don't get gene expression. So the operon is off. Okay. Green. When there's lactose present in the cell, allolactose is produced. Allolactose binds here, which actually stops the promoter, the repressing molecule from binding. So we can't bind it anymore. But because there's a mutation in LAC-P, RNA polymerase still cannot bind. Even though there's lactose around and our operator is open, we still can't bind here. So we won't get any gene expression because RNA polymerase is unable to bind to the promoter and promote gene expression. So the operon is off, even though lactose is present. Let's keep on the spot. Okay. Okay, now which two regions should I mutate simultaneously? And then we'll talk out the logic of what happens. And then I want to show you what Brooke did last night, or at least probably all weekend, because it was incredible. Which two genes should I mutate? P and Y. All right, Josh. So we're going to mutate P. Ah, no. Stupid. Okay, we're mutating P. So now this DNA sequence is different. So now RNA polymerase can't bind here. And we're also going to mutate Y. So when there's gene expression allowed, we won't get any Y permease protein being made. Lactose won't be able to enter the cell. So now let's look at when lactose is present. No, sorry, when lactose is absent. Sorry, no lactose present in the cell. That means our inducing molecule is allowed to bind to the operator, which will stop gene expression because there's a repressor in the way. So the operon is off. Now let's add lactose. That means allolactose is being produced. Allolactose will bind to the repressing protein right in the same spot that it likes to bind to the operator. So it will not be able to bind to the operator. It does not bind here. Oops. That means RNA polymerase would be allowed to bind to that promoter and promote gene expression, except it can't, because it can't bind to a sequence it doesn't recognize. And so even though Y isn't working, expression of the entire operon is off because the promoter is not working. Okay. Now let's introduce the partial diploid. Now, it's impossible to memorize all possible combinations of mutations. Don't do it. Don't memorize, oh, the promoter does this. The promoter mutation always does this, because sometimes mutations behave differently if they are interacting with other mutations. It's called epistasis. Remember, gene products interact with each other. So don't memorize your way through it. Instead, understand how the lac operon works and apply what you know about that understanding to different mutations. So you should be able to talk this out. So mutate each gene one at a time. Mutate different combinations of each gene one at a time, or two at a time, and try to see how those different mutations would interact. And then add the partial diploid scenario, where you were complementing on a plasmid other types of combinations of mutations. Okay, so here we have on the genome is this one up here that I've been drawing the whole time, and here's our plasmid. On the genome, LAC-I is mutated. So now let's mess up the genome. So LAC-I is mutated, transcript is not made, we don't make this protein, or we make a toxic protein that doesn't function properly. We also have LAC-Y mutated, so LAC-Y is busted. So when the operon is allowed to be on, Z and A will be made, but Y will not. On the partial diploids, on the plasmid, everything, so the I is working, but the P is not. So polymerase will be able to bind to this promoter, but will not be able to bind to the plasmid promoter. And then we have LAC-Z is also mutated. Oh, this feels familiar. I might have done this in class, but I have the other ones also. Okay, I wanted to spend the most time in the chi-square and the lac operon, and then I can get into some of the other things too as well. Okay. Okay. So in the case of no lactose, so lactose is absent, what we should see is the, oh wait, I forgot to express my repressor off of here. Okay, no lactose present in the cell. What should happen is that the repressing protein should bind to the operator, but this one can't do it. It's broken. So it can't bind to that operator. It also can't bind to the operator over on the plasmid. However, the plasmid repressing protein is working fine. And because it's a protein that floats around the cell and looks for DNA to bind to, it will bind here to this operator and block gene expression off the plasmid. It will also bind to this operator and block gene expression off the genome. And so because we still have this working repressor, even though this one over here from the genome is busted, this one is working. And so the operon is off and you get no gene expression like you normally would. And so what that means is the mutation, the transacting protein here, which is, it just means that the transacting is the protein can float around and bind to any DNA element it recognizes. That is easily complemented by a working copy of that protein coming in on a plasmid. So we can complement the LAC-I mutation on the genome. Now let's add lactose. We're making allolactose. So when we're making allolactose, that will bind here, but I mean, like, it doesn't matter because it's broken anyway. So it is going to continue being unable to bind to the operator. But allolactose will also bind here. And so the repressing protein will now not be able to bind to the operator on the plasmid. It also will not be able to bind to the operator on the genome. So all repressor proteins are deactivated. That means RNA polymerase can bind here and express genes here, but we're only going to get Z and A. We won't get any Y because the Y on the genome is busted. So let's see if we can complement it by a working Y on the plasmid. So RNA polymerase binds to this promoter. It's also going to try to bind here, but oh, that one's broken. So RNA polymerase cannot drive expression off of this plasmid-borne one. And so even though Z is broken, it doesn't really matter. You wouldn't get Z anyway because this promoter is broken. You're also not going to get Y here because you cannot, because this regulatory element, the promoter on the plasmid, regulates everything it's attached to. So having a working copy of the promoter over here in the genome does not complement a broken promoter over here. It doesn't float around in the cell. It's physically attached to these genes. So it can promote expression of anything it's attached to, these ones, but it cannot promote expression of genes found elsewhere, like on a plasmid or someplace else in the genome. So that means in the presence of lactose, you'll get Z, but you won't, and you'll get A, but you won't get Y, because even though it's working here, it can't get expressed, and it's not working here, so it won't get expressed from the genome. So if this is Y, ZY, this is lactose minus, this is lactose plus. This would be no expression of these when there's no lactose because we're still able to repress the working repressor. And then when we turn it on, lactose is present. We will get Z coming from the genome, but you won't get it from the plasmid. And you don't get Y in either case because it's broken on the genome and not expressible off the plasmid. Want me to do another one? Of the diploid one? Partial diploid. Okay. Brooke says sure. Oh, Renee says yes. Okay. You got it. Let's look at this one. Okay, so I got my genome. So let's start to mutate some stuff. All right, so on the genome, we have a mutated promoter. That's going to be a problem. We also have a constitutively active operator, which means this is also busted. Oh, this is an interesting one. Okay, and we have no LAC-Z coming from the genome, and our LAC-Y is working fine. On the plasmid, we have no LAC-I, so this LAC-I is mutated. Oh, these three are mutated. And so we don't get any of this. We don't get any working I from the plasmid, but our I is still working in the genome. And then everything else in the plasmid is working except for Y is not working. So when lactose is present, and we should get Y from the plasmid, we won't because it's mutated. So we'll just get Z coming from the plasmid. This is very interesting. Let's work it through. Okay. All right. Let's add lactose to the system. So I'm going to put that over here so this is clear. And this is not plus, this is minus. So no lactose in the system. What's supposed to happen? Well, our repressor should bind to the operator. Ooh, but the operator is broken. So it is not able to bind to the operator. Its sequence has changed, which means we cannot repress this operon anymore. And so for this one, this repressor is busted. So it can't bind here and it can't bind here. So we don't even have to consider what's going on there. But we do have a working one coming from the genome, and it can bind to this operator and block gene expression from the plasmid. But it can't bind to this operator to block gene expression off of the operon. So what we would expect to see is RNA polymerase in the absence of lactose, which is weird, binding to the promoter and promoting gene expression. And then you would expect to see RNA polymerase trying to do that here too. But because there's a repressor off the plasmid, it's off over here. And then because the operator is open, it should be on, but the promoter doesn't work and RNA polymerase can't bind here. And so that is actually dominant over a constitutively active operator molecule. So even though a constitutively active operator on its own would mean this operon is on all the time, the promoter is broken. And so that means on the genome, a LAC-P mutation is dominant over a LAC-OC mutation. Both exist on the genome, and the LAC-O mutation would basically make this constitutive and turn it on. A LAC-P mutation would keep it off. And because RNA polymerase can't bind and promote gene expression, it's off, man. It's off, baby. There's no way to turn that thing on from the genome. So you're not getting it off the plasmid because the regulator is bound to the operator and RNA polymerase can't promote gene expression. You're not getting anything from the genome because the promoter in the genome is broken. Even though its RNA polymerase is not blocked, it also can't bind. That's a huge problem. So you would get nothing in either case. Now let's add lactose and see what happens. Okay, adding lactose will stop this repressor from doing its thing. And it doesn't really matter. This one's already not doing anything, but we'll add it anyway. So blocked, blocked, blocked anyway, because it can't bind to the operator there anyway. But now also blocked there, can't bind to the plasmid. Okay. So now RNA polymerase should come in and express, and it does. We get expression off of this one because the promoter is working, and we get Z. Oh, but Y is busted, so we don't get any Y coming off of here. Nothing, no Y there. So we definitely get Z, and that will be present in the cell, being expressed off the plasmid. We can't get Y off the plasmid, so let's see if we can get it from the genome. Okay, so we got rid of that repressor molecule. It's not binding. It wasn't actually binding to the first place. But remember, RNA polymerase, if that promoter is mutated, still cannot bind, and we still are off, and you still get no gene expression. This is all off because your promoter is not working. And so even though Y is working on the genome, you're not ever going to see it. It will never be expressed because its regulatory element is broken. And so it's minus there. properly executing this depends critically on your understanding of what is expressed when lactose is absent and what is expressed when lactose is present. It critically depends on your understanding of what a structural protein is, what a regulatory protein is, and how those are different from regulatory elements that do not make proteins and don't float around the cell, but control whether or not structural proteins are on or off. If you understand all of those, any combination of mutations, whether single or double, or complementation with a plasmid, you can figure that out, as long as you understand how the system works when everything is working normal. Questions? Okay. It's freezing in my house. I'm so cold. Okay. All right, I haven't moved. I've been sitting here for hours talking to you guys. Oh, I haven't moved around. All right, let's talk pedigrees. When you, so on the exam, you'll either be given a pedigree to interpret or you'll be asked to draw a pedigree from a description. So you need to be able to do both of those, and both of those examples are in the problem set. But just to remind you what types of patterns you are looking for, you have your choice of 4 options. You have autosomal recessive, autosomal dominant, X-linked recessive, and X-linked dominant. And when you are trying to interpret a pedigree, start with the question, is it dominant or recessive? Once you can come up with a pattern associated with either of those modes of inheritance, then assume you are absolutely correct, and then try to determine, given that I assume it is recessive, is it autosomal or X-linked? And if you do it in that very specific order, you are more likely to come up with the most likely mode of inheritance, as opposed to just going crazy when you are trying to assume, or you're trying to apply all 4 at the same time. So go in that very specific order. And this is what you're going to look for. For recessive traits, individuals must have both alleles to actually have the phenotype. So that means individuals that are affected, it's possible for neither of their parents to be affected. Individuals who are affected, it's possible that neither of their parents are affected. And so because of that, the trait tends to skip generations. It's absent from this one, not here, but it suddenly shows up in generation 4. So it tends to skip generations because it hides in the heterozygote classes. So in order for individual 2, 3 to be affected, this person must be a heterozygote, this person must be a heterozygote, and this person is homozygous recessive. Okay, that's the biggest one that you're looking for recessive traits. This also means that 2 affected parents will produce 100% affected progeny, because for 2 parents to be affected, that is this cross. And so they're only ever going to have one type of progeny, which is affected. Also for recessive traits, there can be carriers. So individuals can be heterozygous for a trait, and they are showing the dominant phenotype and carrying the recessive one. So they themselves are unaffected, but they can give rise to affected progeny if they meet up with another carrier. For dominant traits are every affected parent, every affected individual has at least one affected parent. Every affected individual has at least one affected parent. Every affected individual, one affected parent. It does not skip generations for this reason. It tends to show up in every generation. And if it's suddenly missing from a generation, it will never appear again for the remainder of those progeny, that family lineage. So it does not skip generations. You only need one allele to express the phenotype. So these individuals are either homozygous dominant or they're heterozygous. That means unaffected people are only homozygous recessive. There are no carriers in a dominant trait. You can't carry a dominant allele without expressing the trait. So you're not a carrier if you're expressing the trait. That's how you separate dominant and recessive. Now, how do you go with autosomal. Well, once you assume my trait is recessive, then you start to look for patterns associated with autosomal inheritance, given that is also recessive. So autosomal inheritance, usually both sexes are affected equally. And you can get transmission of the trait from father to son, father to daughter, mother to son, mother to daughter. It doesn't matter because it's not being inherited on their sex chromosome. Compare that to X linkage, which is a little bit different. So for X-linked recessive traits, affected sexes tends to be infrequent. It tends to be uneven. So for X-linked recessive disorders, more females are, no, sorry, gosh, I'm so sorry. That's because I've been talking for too long. For X-linked recessive traits, more males are affected by these traits. More males are affected because females can be carriers and they can be carrying the allele. But if they are heterozygous, then they're expressing the dominant allele here and only carrying the recessive allele. Whereas males are either going to be carrying the dominant allele and showing the trait, or having the recessive allele and showing the trait. In either case, they're going to express whatever allele they have. There's no carriers for a recessive trait in a male. It's not possible because there's only one X chromosome in a male. This means that they cannot be passed from father to son. And affected men produce 100% carrier females, and affected females will have 100% affected sons. So that's why the distribution of the trait is uneven between males and females, and more males tend to be affected because it's going to show up in any male with a trait and not necessarily in females that have the trait. Compare that to X-linked dominant traits where more females tend to be affected, because there are two genotypes where you're going to have an affected female. So that's 60% of genotypes, right? 2 out of 3. Compare that to males where only 50% of the males will have the trait. So it goes down 10% because there's only 2 possible genotypes, one of which is affected and one of them isn't. Also, if it is at all lethal at all, it's going to slightly more females are going to be affected by that. Or sorry, so if it's all lethal, it's going to kill the percentage of males that actually carry the trait. It might not kill it, so it might kill in this female, but not necessarily kill in this female. So it's more females will be born with a lethal X-linked dominant trait than males will be. So you tend to see it more in females for that reason too. Affected males will pass it to all of their daughters because affected males look like this. And they have to pass their X chromosome to their daughters, and they only have one X chromosome to give. It's the affected one. So 100% of an affected male, 100% of daughters from an affected male will have the trait and show the phenotype, but none of their sons will, because they're not giving X to their sons, they're giving Y to their sons. So those are the real dominant patterns that you're looking for. Let's apply. Okay. Determine the mode of inheritance for this pedigree. So we make a couple of assumptions when we do pedigrees. We assume the trait is rare. So people marrying into the family tend not to have this trait. Not always, but tend not to. And then it's fully penetrant, meaning individuals that are affected are going to have the expected genotype. So if this is a dominant disorder, then they have at least one copy of the allele. If it's recessive, then they have 2 copies of the allele. It's fully penetrant. So anybody who's affected has the expected genotype. So let's start first with dominant or recessive. What patterns do we see that support either dominant or recessive? Guys, see anything? What do you see? Anybody here? Yep. Oh, we got one more. Gwen says recessive. Gwen, why? It skips generations. So let's confirm that for her. Okay. So in this generation, we see, oh, I knew that was going to happen every time. Now, is it dominant or recessive? Will be in red. Well, it shows up here. It skips here. It skips here. It skips here. But it also doesn't show up here. So that doesn't really help us, right? If we look at that family group. But let's look at this individual. This individual is affected and both parents are unaffected. There's no way that this is a dominant trait. If it was dominant, this individual 3-5 would have to have gotten the allele from either one of their parents. And if it was dominant, one of their parents would be expressing the mutation, and neither of them are. So it must be recessive. This is also supported by 2-2, who is affected, no affected parents. So it's definitely recessive, for sure. So now, assuming we are correct and it is recessive, which there'd be no other way this would be the case unless our assumptions are wrong, right? That it's not fully penetrant, but don't confuse yourself. So we assume we are correct, it's fully penetrant. We assume we are correct that it's recessive. So now, given that it is recessive, is it autosomal recessive or X-linked recessive? And so look for the distribution of this trait among males and females. Look for the passage of father to son, because remember that an affected father will pass it to none of his sons, and all of his daughters will be carriers as long as the other partner is not affected. So let's look. So we've got 1, 2, 3, 4 affected males that are in the population. And we have 1, 2, 3, 4, 5, 6, 7, 8 males total. So 4 out of 8 are affected. And this is going to be some easy math for the females. There's 0 affected females out of many females. Oh, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 out of 11. So that's certainly trending more that males are more affected, and a recessive trait where males are more affected is more likely to be X-linked. So if we say, okay, it's looking like it's X-linked according to our male versus female data, but in order to make sure that if you say it's X-linked recessive, that it is never passed from father to son, because it can't be. X-linked recessive traits in a man look like that. And so all of the males will receive the Y, all of the females will receive the affected allele, but as long as the other person is not affected, and it's super rare in the population, that person is unlikely to be a carrier, she is probably giving her dominant X to all of her daughters, especially since none of them are affected, right? None of them are shaded in. So we don't see any violation there. There's no passage from father to son here, and that's essentially it. So is it possible that it's autosomal and we just happen to have a string of males that are affected? It's possible, but we're leaning more towards X-linked recessive here. It's far more likely that it's X-linked because there's all these males that are affected, and we don't see it ever being passed from father to son. Let's do one more. Okay, so we assume the trait is rare. So it's rare. Everybody who's affected is going to express the expected genotype. And so now let's start with, is it dominant or recessive? Let's look for some patterns here. Dominant or recessive, what do we think? It's dominant, it's in every generation. Brooke, you're right. It does show up in every generation, but let's make sure that every affected, oh, I almost happened again, I'm so close. That every affected individual has one affected parent. That's true there. 5. Can you tell I'm frustrated? Affected. This person also has one. Both of these are affected. They have an affected mom. These are both affected by their dad. So it's showing up in every generation. It doesn't show up here, but it never shows up here. Note how it's never there. And so when you see that, that's not saying it skips a generation. That's essentially saying nobody inherited that dominant allele. And so it's just not present in their family lineage. However, 2-5 inherited the dominant allele and passed it to 3-8. 3-8 inherited the dominant allele and passed it to 4-5. And so every affected individual has at least one affected parent. It's certainly not skipping generations when you follow one family lineage. So it's very likely to be dominant. Is it possible that it's recessive? It's possible. It's just not very probable. And we're trying to determine the most likely mode of inheritance. It's a hypothesis. It's an educated guess. Without going in and sequencing all of these people, that uneducated guess is the best we can do. So now, given we assume we're correct and that it's dominant, is it autosomal or X-linked? Take a look. Tell me what you see. I'm going to erase all of my, so it's a little easier. Three out of 10 males are affected. What about females, Brooke? Six out of 13. So 3 out of 10 males. Oh, I don't know. I don't know. I haven't done this one yet. But let's assume you're fine. 6 out of 13 females. And so what's this? This is like 30%-ish, 33, 30%. 6 out of 13 is 46%. So we have slightly more females affected. Slightly more females being affected tells us that it could be X-linked dominant. But let's make sure that you don't make any violations for X-linked dominance. So Marion is saying, I think it's X-linked because the fathers pass it to all their daughters, affected fathers. So let's see, we have an affected father here, and his daughter's affected, his daughter's affected. We have an affected father here. Oh, pause for Christmas. Okay. I don't know how long this one is. It means I've been talking for, wow, a long time. Is it cute? Oh, that was a short one. Okay, good. My kids love that clock. They get so pumped every time I take it out of the basement to put it up on the wall. And like my husband and I hang it like, oh no. And we were going to be tortured every hour, but it's cute. Some of them are long. Some of them are like a full send on the song. And it's like, when is this going to stop? But both kids, the second that clock starts, they break into dance. And it's pretty cute. They enjoy it. So I'll put it up for them. What are we doing? So affected fathers pass it to all their daughters, affected fathers. So that one is certainly true. So now we have two patterns associated with X linkage. More females are affected, and affected fathers pass it to 100% of their daughters. They should also pass it to 0 of their sons. 0 of their sons, 0 of their sons. So that's looking pretty good. Let's see, let's think about affected moms. So affected moms are going to be, so this mom is a heterozygote if it's dominant, because she got a good A from dad and a bad one from mom. And so we could potentially have a mixture, but unfortunately both of these individuals received the bad one from mom. I mean, I'd say it's pretty good. I'd say I'm leaning more X-linked also. Again, it's possible that it's autosomal, but we've got two evidence patterns that say that it's more X-linked, more likely to be X-linked, and that is more females are affected, and fathers pass it to 100% of the daughters in 2 cases and none of their sons in 2 cases. So I would go with X-linked on that one. Support your answer. Support your answer with evidence. Questions? I have energy to do another half an hour. You guys with me? You've been here just as long, especially if you're in biochem. Do you want me to keep going? I can keep going. Yes.
please.
Yes, please. Yes, keep going. Okay. All right. No problem. If I start to go, like you start to see me go crazy, then you know why. Because I've been talking since 10:30. Renee is asking me to go over DNA sequences. Well, what a great segue, Renee. You're welcome. Happy to do it. All right. Yeah, no, of course. You're welcome. All right. So for mutational analysis, here's the things you need to make sure you can do. Oh no, I'm going crazy. Come on, pen. Okay. You need to be able to express a gene. I hate to go. Thank you for the review. Oh, no problem, Brooke. Oh, Brooke, I didn't even get to brag about the amazing work you did. Hold on a second. Let's like, you guys got to see this. Okay. So Brooke worked on, she worked, oh, stop. Okay. Where is it? I think, oh crap, it's not on this computer, Brooke. It's so sad. Wait, it's got to be in my inbox. She has been working on creating, she doesn't get to use a cheat sheet, but she certainly made one. She went for it. Was it you? It was you, right, Brooke?
I think it might have been the other Brooke.
Oh, oops. Really? Okay, wait. I get that.
Maybe.
I get that Brooke here too. It was the other Brooke, Brooke Ryder. So sorry, Brooke. Oh my God. How many times am I going to confuse you guys? I'm so sorry. Okay. She created this I highly recommend for if you, I say do something with this information, do something with this information. Look at this gorgeous cheat sheet that Brooke Ryder made. So she took everything that was on a slide from all modules and squished it on to one giant cheat sheet. So you can zoom in and review all of the slides at once for any topic. So she's got all of module 1 over here, right? So transformation principle, Char-Gas rules. In the middle here, we have mitosis, meiosis, and mod 2, pedigree analysis, which goes all the way down. Everything that I wrote on a slide, she put on this cheat sheet. And so by doing that, by trying to make those connections and putting it all in one place, all of this stuff is probably also in her brain. Isn't this incredible? Look what she did. She drew all of these out instead of actually copying and pasting the images. She drew everything out, drew out the cell cycle. Gorgeous, incredible work. Look at that, everything. So she is certainly thinking about genetics. She's definitely prepared. So nice job, Brooke, when you eventually watch this later. Just wanted to showcase that because it was pretty awesome. Okay. All right, back to my thought. Okay. So when you're doing mutational analysis, you need to be able to express a gene. What that means is you need to be able to turn DNA into RNA, which is transcribing your gene. And you need to be able to turn RNA into protein, which is translating your gene or an amino acid sequence. I really shouldn't write amino acid because it's a sequence, it's a polypeptide. Those are amino acids held together by covalent peptide bonds. So you need to be able to do that. Given that, you need to be able to mutate a nucleic acid, knowing that mutations that matter, mutations that are going to affect the protein, happen in the template DNA. Template DNA. That is your DNA and your double-stranded DNA molecule. That is the DNA strand that runs 3' to 5'. That's your template DNA. How do I know that? Because when DNA runs in a 5' to 3' direction, 3' to 5' direction, RNA polymerase binds to that and will make RNA starting with the 5' end, which is antiparallel and complementary to the 3' end of the template, and finishing with the 3' end. So that's how you know the template strand is the 3' to 5', because the RNA that's complementary to it runs anti-parallel and complementary to the template strand. And then when the ribosome binds to the RNA, it starts to create a polypeptide sequence starting at the N terminus and linking amino acids together until you get to the C terminus. You also will need to classify a DNA mutation as a transition, transversion, or indel. So I put this over here, that if you order your nucleotides, oh my gosh, I'm gonna lose my mind. Don't lose your mind, Ellis, don't do it. If you put your nucleotides in alphabetical order, top to bottom, so A, C, G, T, alphabetical order, top to bottom, so top to bottom, top to bottom. If you write them out like that, everything horizontal is a transition and everything else is a transversion. That's the easiest way to remember the difference. You have to be able to classify a protein change as one of these following terms: a missense, which is something like one amino acid changes to another amino acid. So that's a missense mutation where the amino acid actually changes as a consequence of the DNA changing. And you get missense mutations from transitions and transversions. Those are base substitutions, and only base substitutions will change a single amino acid. If you see multiple amino acids changing, that is called a frame shift mutation, where all of the amino acids and like the second half of the protein are completely different. And that is always caused by an insertion or deletion. So missense, nonsense, neutral, and silent are all caused by base substitutions, which are transitions and transversions. Frame shift mutations are always caused by indels. A nonsense mutation is when you go from a sense codon, something that is encoding for an amino acid, to a stop codon. And so you're missing that amino acid and everything else that comes after it. A neutral mutation is when you swap out say a small nonpolar amino acid for another small nonpolar amino acid. They're chemically very similar. It is a missense mutation. You do get a change in amino acid sequence, but because they are so similar chemically, the protein will fold normally and function normally. And so it's called a neutral mutation. There is a mutation at the amino acid level, but it's neutral. It actually doesn't change how the polypeptide functions. A silent mutation is no change at the amino acid level due to redundancy in the genetic code. So we do see a change at the DNA level, a transition or a transversion, but the amino acid remains the same. And so the protein doesn't change, functionally speaking. Okay, you also need to be able to use a mutated amino acid sequence to predict the change at the DNA level. But I won't give you an amino acid sequence and tell me the entire DNA sequence that made it, because that would be impossible. There's too much redundancy in the genetic code to do that. For example, there are 6 codons that encode for arginine. So if you had arginine in your polypeptide sequence, you could predict that it's one of these 6, and you could probably tell me that the middle position is G, but that's all you could really tell me. However, if you see an arginine change to a serine, so that's a missense mutation, you can probably predict that one of your arginine codons, you had a single base substitution here, which resulted in the codon changing and the amino acid changing. So that would be a missense mutation caused by a base substitution. But you couldn't know for sure if it's CGU to AGU. It could have just as easily been CGC to AGC. So you can't tell me exactly what genetic mutation occurred. You could give me a suggestion, a possibility, but you can't tell me that's definitely the answer, because there's just too much redundancy in the genetic code. but you can certainly tell me what likely changed, one of the many possibilities leading arginine to change to serine at the genetic level. Okay, let's apply. Now this one's a little tricky, but let's go for it. We're studying the effects of a toxin isolated from pathogenic bacteria and expression of genes and neutrophils. You isolated RNA, so this is RNA, and how do I know? Because it's got U's in it. See all those U's? That's RNA. Using RNA sequencing technology, obtain the following sequence from your gene of interest. What is the sequence of the template DNA strand that was used to produce this RNA strand? The template DNA strand means that's the 3' to 5' anti-parallel complement to this RNA strand. So how are we going to do that? An emergency. I will return. Let me finish this thought. Okay, I'm back. All right, so how do we complement the template strand? Since this is given to us in the 5' to 3' direction, we can determine the template DNA strand as the anti-parallel complement to this RNA. So that's going to be TAC, TCA. Remember, for every U that's present, there was an A in the DNA template. And for any A in the RNA, that's going to be T in the DNA template. Because T's and U's are replaced between DNA and RNA. This is going to be T, G, C, G. Okay, and then any mutations will be in the template DNA. So that's the sequence of the template DNA. So let's also do the amino acid sequence of the protein that's encoded by it. I wish I had. I don't. All right. Well, I know AUG is methionine because that's the only start codon. I know UAG is a stop codon. And that's it. So now we got to figure out AGU. You know what I'm going to do actually? Just to make this go a little easier. Okay, AG, oh, Alexa, you're in the way. Hold on, I'll move you. AGU, AG, U, serine, GGA, glycine. Okay. There we go. So that's gene expression, transcription, translation. I actually did reverse transcription there as I went from RNA to DNA. Okay, what is the altered amino acid sequence of the protein given the following mutations? And then what is the phenotypic effect? So that means what happens to the protein given each of the following mutations. So I've got a transition at nucleotide 4. Oh no, I can't remember which is which. So I will write my nucleotides in alphabetical order from top to bottom, and then all of the horizontal ones are transitions and everything else is transversion. Nucleotide 4 is this one here, the template DNA. And so a transition for T, and the template DNA is T to C. So we're going to go TCA to CCA, which in the RNA is GGU, which in my genetic code table is glycine. What used to be there was serine, and now there's glycine. What do we call that at the protein level?
Missense.
Yes, thank you. Wow, Joanna, you're still here. Are you so tired? Okay. It's a missense. Because the amino acid changed from serine to glycine. It went from one sense to a different sense codon, but it's a mistake. It's not supposed to happen. It's a missense protein mutation. Okay, a transversion at nucleotide 7. And I have to say, I said here, give all possible answers because this was a practice question from the study guide, I believe. I'm just going to do one just to illustrate my point. So a transversion at nucleotide 7. So we have nucleotide 3, 6, and 7 is here. It helps to split your nucleotides into sets of 3, so it's just easier to count that way. Also, sets of 3 means they're codon, so you can sort of visualize a little better. A transversion at nucleotide 7 is a C. So a transversion can either be to G or to A. I'm going to do A. And so we go from CCT to ACT, which in the RNA is UGA. Oh, I don't even have to look at my code. That's a stop codon. UGA is the stop codon. It's right here. What do we call that, Johnny?
Nonsense.
Nonsense. That's right. We go from a sense codon, it encoded for something, it encoded for glycine, to a nonsense codon that encodes for nothing or stop codon. And so the amino acid sequence would be MET, SEER, and that's it. You wouldn't see anything else beyond that. And so you're on a test, you will be asked to write the altered amino acid sequence, not writing it out for the sake of time, but you should definitely make it a habit. I'm just showing you the action, what's actually happening. A transversion at nucleotide 9. So now we have 3, 6, nucleotide 9 is here. So we go CCT, and let's do the same transversion. We'll do, oh, wait a second, we can't because it's a T. So transversion is T to G or T to A. So we'll do T to A. That's going to be CCA in the DNA. In the RNA, that is GGU, which is glycine. So we went from glycine to glycine. What kind of mutation is that?
Silent.
Silent. Dwani, why is it not a neutral mutation?
Because the phenotype, like the, oh yeah, like the phenotype has not been, like the function of the protein has not been altered.
So the function of the protein is not altered in either a silent or a neutral. Neither, in both of those cases, the function of the protein is not altered. But what about the amino acid order? Is that different between a silent and a neutral mutation?
Yeah, like the sequence for a silent mutation has not been affected, but if it's the other one, then it's altered.
Yes, correct. So in a silent mutation, the polypeptide sequence, the amino acid sequence, is exactly the same as the mutated, as the wild type. They're exactly the same. There was no change. But in a neutral mutation, the sequence of the mutated polypeptide sequence is different, but the function of that protein has not changed because we switched it from one small nonpolar amino acid to another small nonpolar amino acid. So the function does not change. The sequence is different. The function does not change. For neutral, the sequence doesn't change and the function does not change. That's silent. Okay. The insertion of TGA downstream of nucleotide 12. So let's see. 9, 10, 11, 12. So we are inserting TGA here. That's going to encode for ACU in the RNA, which is threonine. So now the new polypeptide sequence given this insertion is going to be met, ser, gly, arg, threonine, glycine, histidine. What do we call that at the protein level? Well, let's look and see what changed. Methionine is exactly the same, serine is the same, glycine is the same, arginine is the same. We added a threonine, but lysine, histidine are still there. That is an in-frame indel. We inserted a whole codon, so we did not shift the reading frame. Everything else reads the same. We just inserted an amino acid right in there, and we added an amino acid because we added a whole codon, 3 nucleotides. That's an in-frame indel. It's an indel, it's an insertion specifically, but it did not shift the reading frame. It just added added an amino acid at that position, in frame. Compare that to the last one here, deletion of nucleotide 15. So deletion of nucleotide 15, this is 3, 6, 9, 12, 13, 14, 15. That's going to delete this one here. So if we delete that, that means all of this is going to shift over by 1 nucleotide. And so it's going to be TTG, TGA, and then TC something else. So that shifts every amino acid after the point of deletion. And so when that happens, all of the amino acids downstream of that deletion after the deletion are going to be different. That's your frame shift mutation. It shifted the frame, and so multiple amino acids have changed instead of just inserting one and everything else remaining the same. This is your study guide, so if you want to draw that out for yourself, I highly recommend doing that. Study guide for mod 7. Okay. I'm just seeing what I have left because I'm going a little crazy, but that's okay. I'll go just a little crazy for you guys. That's fine. So an inbreeding question. In order to answer any question in population genetics, you need to be able to identify why you would use each of these equations. So I want to do that for you right now. So this is the equation bank. How do I identify when I use which equation? For this one here, what? No. This is observed. No, it isn't. This is just allele frequency. This, because it's divided by 2n, that is observed allele frequency specifically. Once you find the observed allele frequency for big A, you can then apply this one, this equation here, to find the allele frequency of the other allele, little a. Or if you're given, saying like you assume the population is in Hardy-Weinberg equilibrium, the frequency of P is 0.4, then you can apply this P plus Q equals 1 to know that the other allele frequency would be 0.6. But this very specifically, because it's divided by the number of individuals carrying those alleles. This is observed allele frequency. This is observed genotype frequency. This, however, is expected genotype frequency. You use this if the population is in Hardy-Weinberg equilibrium, or you expect it to be in Hardy-Weinberg equilibrium, or if you are testing the null hypothesis that it is in Hardy-Weinberg equilibrium and you need expected values. Okay, this one right here has M in it. That is a migration coefficient. This is your migration equation. And you recognize that by the presence of M being your migrants, percentage of migrants coming from the source, and then the percentage, and then 1 minus that is the amount of residents that were in the recipient population. All of these are inbreeding equations. And so you can, given the, if you know what the inbreeding coefficient is, which is F, you can determine the effect of F on each of these different genotype frequencies. So that is your inbreeding, testing for inbreeding. A lot of people used that on the exam. I don't really know why. I have a suspicion. I suspect that you have copies of past exams where I have used this before and not migration. And so you just assumed it was the same question, and it's not. I don't ever write the same test twice, ever. And then all of these, that's a suspicion. I don't know if that's the case. It's just that it's a guess. All of this, the rest is all natural selection. Testing the effect of fitness on a population. Why? Because fitness is showing up all over the place in these equations. So that is what you would use to test for natural selection. In rhododendron flowers, color can be determined by 2 codominant alleles, big R and little r. Remember that codominance is, we see the phenotype in the dominant as red, we see the phenotype in the homozygous recessive, which is white, but in the heterozygote class, it is both red and white patchworks. So it's not a pink flower, it's a red and white patchwork flower. So that is codominance. Okay, this is a little tricky. Okay, so the frequency of R in a population of rhododendrons is found on a small island is 0.75. You might be thinking, oh, small island, she's probably going to talk about migration, but I did not. I then say, hey, if random mating takes place, that doesn't have to do with migration. That is one of the assumptions of Hardy-Weinberg law, that populations are randomly mating. So if you see the word mating like this, ooh, now we're moving away from something like migrating from island to mainland, and we're talking more about mating, which is going to be the effect of random mating or non-random mating on your population. So if we are random mating, this is one of the assumptions of Hardy-Weinberg equilibrium, that the population is mating randomly. And so we are now allowed to use P squared plus 2PQ plus Q squared because our population is randomly mating. what are the expected frequencies of red, white, and red-white color types? So if the frequency of big R is 0.75, then I know the frequency of little r is 0.25 because these have to equal 1. I'm going to call big R P and I'm going to call little r Q. It does not matter what you choose, you just must be consistent. So 0.275 squared plus 2.75 Q, and then this one is 0.25 squared. That means the frequency of my heterozygote, oh, it's R, right? My homozygote, or P squared, .56, the frequency of my heterozygote, or 2PQ. And then the frequency of my homozygous recessive, or Q squared. That. Okay, so those are my expected frequencies, assuming the population is randomly mating, assuming we are in Hardy-Weinberg equilibrium. Okay. Now, part B of this is if the inbreeding coefficient for the population is 0.3, what does that mean? It means F equals 0.3. It means 30% of this population is inbreeding, finding mates that are genetically related to each other. So that inbreeding coefficient goes from 0 to 1, where 0 is 0% of the population is finding genetically related individuals and mating with them. Or 1, which is 100% of the population is finding genetically related individuals and mating with them. So what are the expected frequencies of these genotypes, considering that there's inbreeding in the population? So that's where you would go to your inbreeding equations. Remember what inbreeding does to genotype frequencies in the population. Does inbreeding tend to increase or decrease diversity? Okay, so spoiler alert. Oh, no. Does somebody get it? Decrease. Yes, Josh, good job. It decreases genetic diversity in a population. And so we're going to decrease. Oh my God. We're going to decrease the amount of heterozygotes, which really represents diversity in a population, the amount of mixtures that we have. And we're going to increase the amount of homozygosity in the population. We're going to decrease the amount of heterozygotes by a factor of F. And we're going to add by a factor of F to the others. Because I have a, Because I have a terrible memory, I'm going to go to my equation bank and make sure that I wrote that equation properly. So yes, plus FPQ, oh, PQ, oh, whoops, I did not write that equation properly. So let's go back. Whereas we are taking one of these heterozygotes and moving it up here, and the other one we're moving it down here. Okay. It's a good thing I checked that. Good thing we have an equation bank. Good thing I don't make you memorize things. I don't care about you getting the equation wrong. I care about you applying the correct equation correctly. Okay, so we expect that this is going to decrease diversity in our population, given that this is where we started over here with these frequencies. we now just plug everything in. And 2PQ, I already know what this value is, right? That is 0.38. Okay. So do we see what we expected to see? Well, we see a, oh, did I have this? Hold on a second. I think I calculated. Did. I wrote 0.5 here and I noticed, well, actually that's less than where I started with the frequency of p squared, which is not what I expected to see. So I either, it's not as, you know, either my assumptions about inbreeding are incorrect or I calculated something wrong. The easier thing to check on that is, did I calculate something wrong? And I did. I didn't add 0.05, I subtracted it by accident. And so I'm going to correct this value to reflect 0.56 plus 0.056, which is 0.62. Okay, so because I went into this problem with an assumption of what inbreeding is going to do to the population, because I understand the concept. I understand that inbreeding is going to decrease diversity and increase the amount of the homozygote classes. After I did my math, I was able to catch my mathematical error because I saw a decrease in a homozygote class. And I know that's not what's supposed to happen. You're supposed to increase the homozygote classes. So my knowledge of the concept helped me catch a mathematical error. This is why you don't memorize equations and ways to do this, right? Understand the concept and you can apply that to any problem. So I went up from 0.56 to 0.62 for RR, big R, big R. For little r, I went from 0.06 to 0.116. So both homozygote classes increased. And then I see a decrease from 0.38 to 0.27. So I decreased diversity, decreased the amount of heterozygotes in my population, and increased the amount of homozygotes in my population. Okay, I'm exhausted. I've been talking for way too long, so I'm going to end with this. To help you with chromosome counting during different cell cycle stages, you need to review the cell cycle and you need to understand what happens at each of these in a diploid state. So I would attach diploid numbers. So assuming my diploid number is 8, and what do my chromosomes look like? They look like sister chromatids. And what happens here? Oh, the cell is growing. Okay, because it just exited cytokinesis. It's building up cytoplasm. It's building up kinases. It's building up templates to those kinases, headed towards the G1 checkpoint, or it enters G0 and doesn't divide anymore. That's why it would enter G0. It's not dividing anymore. But should I decide I need to divide again, I can exit G0 and then I pause at this G1S checkpoint. This is the committed step of the cell cycle. So if I go past this checkpoint, I will finish all the way through to cytokinesis. But these other checkpoints will determine how quickly I do that. Okay. In S phase, my DNA goes from single chromatids to sister chromatids. And so because everything is joined at the center by a centromere, this goes from one chromosome and still one chromosome, but we go from one DNA molecule to two DNA molecules down here. So at G2, my 2N equals 8, but I now exist as a pair of sister chromatids, so my DNA molecules are 16. Check to make sure I have replicated everything properly for my template DNA, and then I enter mitosis. If I am a body cell, a somatic cell, then I'm going to start diploid, 2 copies of every chromosome, and I'm going to finish as diploid, 2 copies in a daughter cell of every chromosome. It's just that I go from diploid sister chromatids to diploid single chromatids, but these are still one chromosome. The genetic consequences of mitosis are I go in as 2N, I exit as 2N. I start with sister chromatids, but I separate them during anaphase, and I end with the same amount of chromosomes, but half the amount of genetic material. Notice in prophase how everybody's lining up, or metaphase, how everybody's lining up end to end like this, as a pair of, sister chromatids in single chromosomes line up end to end. In meiosis, I'm going to start out 2N. I have 2 copies of every gene, right? 2 reds and 2 blues, but I end as an N cell. I only have 1 copy of each chromosome here. Look at this recombination that happened here, so it's a little confusing, but I have 1 copy of the small one and I have 1 copy of the larger one. Whereas here I had 2 copies of the larger one and 2 copies of the smaller one. Wow, that's pretty okay. So I start out as 2N as pairs of homologous chromosomes, each with sister chromatids, and I end with only 1 of each type. Only 1 chromatid. Look how metaphase 1 differs in meiosis from mitosis. Instead of lining up end to end, we are pairing up and experiencing homologous recombination. In metaphase 2, that's when we switch back to end, lining up end to end, but we only have 1 copy of each chromosome in this cell now, instead of 2. So we go from meiosis 1, we start as 2N. When we finish, we end as an N cell. When we start meiosis 2, we start as N and we finish as N. Okay. So understanding that those differences and that concept will help you determine the number of chromosomes per cell, depending on which stage of the cell cycle you're in, or the number of chromosomes and DNA molecules per cell, depending on which stage of M you're in, meiosis 1, meiosis 2, or mitosis. Germ cells do meiosis. Every other cell in your body does mitosis. Remember that we can alter the ploidy of a cell by adding or subtracting single chromosomes from a pair. So in monosomy is one in which we lost a single chromosome. So our diploid count of the cell goes from 2N to 2N minus 1. One of our pairs is missing. One of our pairs of chromosomes is missing its pair. A trisomy is a gain of a single chromosome. So one is got an extra pair. So here it's, let's say we have, my chromosome drawing is getting pretty messy. 2n minus 1 would be missing one of those. 2n plus 1 would be 3 of them in a single pair. A tetrasomy is you gain 2 chromosomes, so gain of 2 homologous chromosomes. So we have 2 sets of triplets here. I'm tired, sorry. So it would be this, but that happening in 2 pairs of chromosomes. Nullisomy is when you lose an entire homologous pair. So it's just like you don't have any chromosome 2s at all because both are missing. So this would be for chromosome pair 1 and chromosome pair 2, you have an extra player in each of those sets. For a tetrasomy, for a trisomy, just 1 set has an extra pair. For a monosomy, that is 1 set is missing its bud. And for a nullisomy, it's both members of a homologous pair are missing entirely, so no. From someone. And this is wild type. Pictures help. Remember that aneuploidy and sex chromosomes is pretty well tolerated. And that's because of X activation. Every single time you have any cell with more than 1 X, one of those is deactivated, squinched up into this tiny little ball of heterochromatin that is never expressed. And so this cell might have this one squinched up and never expressed. And for the neighboring cell, maybe this one is squinched up and never expressed. But because cells exist as tissues, you're going to see a heterozygote in that case. Like both of them will be expressed in the tissue, just not in any particular one cell. And so Klinefelter syndrome is tolerated because this one's going to get deactivated. That machinery exists in males and females. And so they are genetically male because we are expressing everything from those X and Y. And sometimes it can have, but this person's genetically male, phenotypically male person might have some female sex characteristics because there is an extra X chromosome floating around the cell. It's just not expressed that much. For Turner syndrome, this is missing a Y chromosome, so they are phenotypically female. They're missing a Y chromosome or they're missing an X chromosome. Doesn't matter, they're only going to express 1 X chromosome anyway. So it's not going to affect anything but fertility. Same for Klinefelter syndrome. It's not going to affect anything except for fertility. However, for these others, Edwards and Pateau, these are autosomes. These are not sex chromosomes. And having extra any autosome, any, is going to cause physical deformities. It's going to cause cognitive deformities in almost every case. You're going to have both of those. And the bigger the chromosome, the more detriment there is to the cell. And so trisomy 18s happen a little bit more frequently. It's 1 in 8,000 live births compared to trisomy 13, which is 1 in 15,000, because most of the individuals that have a trisomy 13 die before they are born. So aneuploidy is very well tolerated in sex chromosomes. Comparing that to polyploidy, that's a change in the number of chromosome sets or genomes. Not adding an extra chromosome or deleting an extra chromosome, it's adding a whole genome. So instead of 2N, we are 3N. This is only common in plants. This is not common at all in eukaryotic cells that are animals, bacteria either. It's really just a plant thing. For example, I was curious, so I was Googling stuff. The strawberry plant genome is 8N. It has 8 different genome sets. Imagine what kind of crazy pairing there is going on in the development of those gametes. It's got to be relatively infertile. I believe the maize genome, corn, is 4N. So it's just very common for, those are domesticated corn, and domesticated strawberries tend to have more genomes. The more wild forms have less because they're not as genetically messed up, because humans are not tinkering with them. It's pretty nuts. It's pretty crazy. I had a friend in grad school who studied strawberry genetics, and I'm like, why on earth would you pick that? How incredibly complicated that must be. And so this is the short answer question from the first exam. Answers are available. Try on your own before looking at the example, before looking at the answer key, to see if you can do it on your own. I'm tapped out. I'm done. I've been talking for 5 hours. I'm so done. 5.5 if you count my meeting this morning at 10:30. So I'm all done. I hope that was useful and helpful. I will post the recording later today. And good luck on Wednesday. Let me know if you have any additional questions. Practice, practice, practice. Get those terms figured out. Pay attention to the things I told you to pay attention to and practice those first. I will see you all on Wednesday.
Thank you so much.
You're welcome.
Thank you. And Dr. Ellis, is the biochem Zoom up yet?
No, because I'm still talking. I'll put it up
Okay.
now though.
Oh, all right. All right. Thank you so much.
You're welcome.
Thank you.
You're welcome.